Dr. Said, allow me to speak in Arabic for a couple of minutes. برحب بكم جميعا في اليوم العلمي ده اللي بتنظمه الجمعيه المصريه لطب السمع والاتزان، وتحديدا لجنه البحث العلمي. النهارده هنتكلم على الجينيتك هيرنج لوس. هقول بس انترودكشن سريعه كده. انتوا عارفين ايه اجمل احساس في الكون؟ مش انك تعشق بجنون زي ما إليسا بتقول. اجمل احساس في الكون فعلا انك انت تشوف حلمك بيتحقق قدام عينيك، انك تشوف الفكره بقت واقع والواقع بقى انجاز. هو ده اللي احنا النهارده جايين عشان كده. We have a dream. We need to study the actual complete profile of genetic hearing loss in Egypt, and we know we can do it with your help. Everyone in their place can help us to do this research and get the profile of genetic hearing loss in Egypt, because we don't have this information, and we cannot plan for gene therapy or any intervention for genetic hearing loss unless we have this map or the genetic profile of hearing loss. So that's why we are here. We will give you some overview about the genetic hearing loss, and then we have to discuss. It is a short discussions aiming to answer two main questions: what we need and how to proceed to achieve our dream or to achieve our goal. So I'll give you some introduction about the genetic hearing loss. Before that, I want to thank our distinguished guest speakers. We have two international and two national distinguished guest speakers. I'd like to welcome Professor Hesham Shaban from Switzerland, our Egyptian star in Switzerland, and we are very proud of him for his great efforts in the field of gene therapy, especially in cancer and longevity medicine, and he will talk about the strategies of gene therapy in general today, inshallah. Welcome, Dr. Haytham, and I also want to welcome Professor Said Safiuddin from France, one of the pioneers in the gene therapy of hearing loss. So specialized in gene therapy of hearing loss, and we will hope he will share his expertise in this field with us today, inshallah. Thank you, Professor Said. Our national stars, Professor Dalia Hassan, of course, Professor of Audiovisual Medicine from Ain Shams University. Thank you for being with us today. And also Professor Sulaf El-Sayed, Professor of and Head of Medical Genetics Department at Ain Shams University. So Ain Shams University is influencing the meeting very, very hard. I also want to thank my colleagues in the Scientific Research Committee, Professor Muhammad Al-Badri and Professor Mustafa Yusuf, and very special thanks to this lady in the middle, Professor Taqwa Gabr. She did a great effort in planning and organizing this event. Thank you, Dr. Taqwa. And now I'll talk about genetic hearing loss. So what's the genetic or hereditary hearing loss? It's the same meaning. So it's an hereditary condition that occurs when one or both parents pass the condition down to their child through their genes. So this is genetic or hereditary. And the incidence of hearing loss in newborns is about 2 per 1,000. And 60% of this is genetic. The overall prevalence of hearing loss in children is 4 per 1,000, and half of these numbers are genetic. So it can occur at any age. Genetic hearing loss can occur at any age. It can occur, of course, congenital, prelingual, postlingual, adults, and age-related hearing loss is also some form of genetic hearing loss. Congenital hearing loss, 60% of congenital hearing loss is genetic, and 20% is cytomegalovirus. So if you can scan for cytomegalovirus and some genetic problems, we can diagnose many of the congenital hearing loss. It's about 80% of the prelingual hearing loss and 50% of the postlingual hearing loss. Of course, we know the types of Mendelian inheritance. This is we don't have time to explain this, but you know the Mendelian rules of inheritance. But I want to talk about two main types of genetic hearing loss. Number one is monogenic hearing loss, when only one gene is responsible for the hearing loss. And the second type is polygenic hearing loss, when more than one gene is responsible for the condition, for the hearing loss. The main difference between both conditions is that monogenic hearing loss follows the rule that H equals D, means heredity equals disease. If the subject has the gene, he will develop the hearing loss, definitely, without any other factors. So this is monogenic hearing loss. The presence of the abnormal gene alone causes hearing loss. And this for all the syndromic and non-syndromic hearing loss. The second type, which is polygenic hearing loss, it follows the rule H plus E equals D. That's heredity plus environment equals disease. So the presence of the gene does not give the condition unless there are some environmental factors. And these types of hearing loss include age-related hearing loss, noise-induced hearing loss, ototoxicity, and Meniere's disease. So these subjects have genetic susceptibility to develop these kinds of hearing loss. What if they are exposed to the environmental factors and don't have the gene? They will not develop the condition. So you have to have the gene and the external factors. The typical example of this polygenic hearing loss is type 2 diabetes mellitus. We know that type 1 diabetes mellitus is an autoimmune disorder. It's not a genetic disorder, but type 2 is genetic. I am a type 2 diabetic, and I have a gene that's responsible for the appearance of the condition if you are exposed to the environmental factors. High glucose intake, stress, obesity, all these environmental factors with the genetic susceptibility will develop type 2 diabetes. So this is the difference. Okay, so we have many types of hearing loss that are genetic, either monogenic or polygenic, including, of course, presbycusis or age-related hearing loss. 30% of the genetic hearing loss are syndromic, appearing not in isolation with other clinical findings. 70% are non-syndromic or isolated hearing loss, and sometimes there is what's called mimic non-syndromic, and this is important for the diagnosis because mimic non-syndromic means that it appears as a non-syndromic and it is syndromic. And the typical example of this is Usher syndrome, Usher type 2, that the hearing loss appears early in life and then later in life the eye symptoms will develop. So if you have this child early in life, you can diagnose it as mimic non-syndromic isolated hearing loss. So eye examination in such children is very important to exclude syndromic hearing loss. Of course, there are more than 400 syndromes that having hearing loss, and only 30% of genetic hearing loss are syndromic. And for the non-syndromic, which is 70%, there are more than 140, of course. Our dear professors will talk about the genes responsible for hearing loss, but more than 140 genes discovered to cause hearing loss. And to name these genes, they have a code. And the code, as you see, it's DFN, stands for deafness, and then A for dominant, and B for recessive. So an X for X-linked. So if you have D, F, and B, this is a recessive, and this is the majority of cases, 80%. And D, F, and A, this is the dominant subtype, and D, F, and X, the X-linked, and we have mitochondria. Of course, these are very rare, the X-linked and the mitochondrial hearing loss. Many names and genes for this many genes, but we can classify the genes responsible for genetic hearing loss into six main types. The gap junction or the connexins, which is important, the GGB2 encodes the connexin 26. and the mutation causes deafness, of course, and it can be dominant or recessive. Tight junction proteins, the claudins, cytoskeletons, the transmembrane proteins, transcription factors, and micro RNAs. These are the types of the genes that can be responsible for hearing loss. But 5 genes are responsible for more than 50% of genetic hearing loss. This is GGB2. It's 20% of cases, and this is a global percentage, the global share of this gene to cause hearing loss. We do not know exactly in Egypt how much, maybe 10, maybe 11, maybe 12. We have some percentage, but it's globally it's 21, STRC 14%, SLC 20, and this 50%, 5 genes only can cause 50%. What does it mean? That if we can screen for these 5 genes, we can at least exclude 50% of the genetic hearing loss. And this is an example how heterogeneous is this genetic hearing loss. The GGB2 mutations are responsible for 50% of autosomal recessive non-syndromic hearing loss in Europe. Globally, we said it's 21. In Europe, it's 50%. And you see, these mutations do not cause hearing loss in sub-Saharan African countries. So they have the gene, the subject has the gene and not hearing loss. So this is how it is heterogeneous, and single gene can cause many types of hearing loss: autosomal dominant, autosomal recessive, and sometimes syndromic also. For this GGB2 is 50% in Europe. If you consider the cochlea as a true hearing aid, that the amplifier is the outer cells, the microphone is the inner hair cells, and the filter is the basal membrane, and the stria vascularis is the battery. And if we know that connexin 26 is responsible for production of ATP in the stria vascularis. So 50% of genetic hearing loss in Europe are caused by a bad battery. Consider this. The cochlea is normal. Only the ATP production is abnormal, and this will cause hearing loss. So we have the clinical aspect. So it can be any kind of hearing loss, any severity and laterality, any configuration, takes any configuration, any course. And this is the rule for the clinical profile of genetic hearing loss. We have autosomal recessive non-syndromic hearing loss. It's bilingual, it occurs bilingual, severe to profound hearing loss, stable, affecting all frequencies. This is the general pattern of recessive non-syndromic hearing loss. For the dominant non-syndromic hearing loss, it's postlingual, mild to moderate hearing loss. It's progressive, affecting a subset of frequencies. Thanks God, this is the minority. This is only 20%, and autosomal recessive is 80%. Why? Because the autosomal dominant non-syndromic hearing loss has a problem. What's the problem? They pass the neonatal hearing screening. They are normal at birth. They have normal hearing at birth. And they pass the neonatal screening. And after that, they develop the hearing loss. Even if they have mild hearing loss at the screening, may they pass the screening. That's why we need to recommend further screening after the newborn or neonatal hearing screening. But this is the rule, and the exceptions are more than the rule. So we can have autosomal recessive with postlingual hearing loss. We can have autosomal dominant with bilingual hearing loss, with different severity, different configurations. So everything is possible here. I will not talk about this too much. These are the common syndromic hearing loss. The differential diagnosis of genetic hearing loss, number one is cytomegalovirus. It's very important cause of etiology of especially congenital hearing loss. We should have a screening for the cytomegalovirus at the time of birth. For the identification, of course, we follow the rule that you should screen before the age of 1 month, you should diagnose before the age of 3 months, and should intervene before the age of 6 months. This is the rule. There are 3 methods to discover gene. Of course, our professor will talk in details about this, but we have the genetic linkage that we do the pedigrees of families and extract the abnormal gene. Tissue-specific approaches, of course, very difficult. The inner ear is not accessible to this. And model system approaches using some animals like the mouse and other. The problem was studying the animals for hearing loss to compare to humans is that the auditory system is different in humans than in animals. I'll give you a various example. The mice, for instance, they are born deaf and they develop hearing in the first 2 weeks of life. For the humans, they hear for the last 3 months intrauterine. So the fetus can recognize his own mother voice at the week 26 of intrauterine. So the humans are hearing for 3 months, 3 complete months. They are hearing fully, the cochlea is fully, the auditory system is fully developed before birth. So this is one of the difference between how to apply this for human. Genetic hearing loss, we can do it, and this one we will have to study this, and we will tell you when discuss how to proceed. Okay, I'm not going to talk about the prevention, but this is important, as we said, that 1 out of 4 newborns, 1 out of 4, 27%, pass initial hearing screening will be later diagnosed as hearing loss or deafness. So neonatal screening is not enough for early detection of hearing loss. Okay, I'll stop at this limit because of the time, and we can discuss this in the discussion area.